Okay, so this is Dr. Clark, and this is part 2 of metabolism, and now we're going into cell metabolism. And so with cell metabolism, there's two different things we look at. One is catabolism, which is breaking down nutrients into smaller molecules. This is usually to produce energy or to make turn a substance into a source of energy. So sometimes like they'll break down proteins to make them a source of energy. Anabolism is using energy to assemble new molecules from smaller components. So this would be like taking amino acids and producing proteins, or this could also be taking something like glucose and actually turning it into glycogen or another polysaccharide. So catabolism has 3 stages. The first part happens within the digestive tract, where food is actually going to be broken down. So that's kind of stage 1 of catabolic metabolism. So the food's going to be chewed up, it's going to pass through the stomach, and the small intestine, large intestine is going to be broken down into smaller parts, absorbed and brought into the body. From there, what happens with the catabolism is that you produce glucose. And so glucose is actually going to first go through anaerobic respiration inside the cytoplasm of the cell. And this is where you're going to get 2 ATP formed, and glucose will actually be broken then down into something called pyruvate. And pyruvate what ends up happening to it is then it goes on the mitochondria, and it's going to go through aerobic respiration with oxygen, and then it's going to produce the extra 36 ATP. So stage 1 of catabolism, you have hydrolysis. The carbohydrates are broken down from polysaccharides into trisaccharides and disaccharides. And finally, those will be broken down into simple monosaccharides. Proteins are broken down into their simple amino acid. Nucleic acids are broken down into their simple nucleotides. And then, of course, fats are broken down into simple fatty acids and glycerol. So once hydrolysis is complete and the nutrient molecules are absorbed by the small intestinal lining, they're going to be transferred into the capillary and into the extracellular fluid, and they're going to be brought into the circulatory system, into the body through capillaries running back into veins, or in through the lymphatic system. So you have lymphatic vessels right here in the duct that they're showing, and then you have the arteries, veins, and capillaries, and all this is going to eventually end up at the liver. In stage 2, what's going to happen here is that those nutrients are going to actually be brought into a cell, and there's going to be an exchange. And so the energy is actually going to be brought into the cytoplasm, where it's going to be broken down, and it's going to produce something called acetyl-CoA and transported through the cytoplasm, where it goes through the 3rd stage of catabolism and the energy source. One thing is when it's right here in this Krebs cycle and it's actually broken down to produce, we call it also pyruvate, but acetyl-CoA is what they have up here. You end up actually getting 2 ATP out of that. And then your 3rd stage right here is aerobic respiration. And aerobic respiration is going to involve producing additional ATP. So you get 2 from this stage right here, and then right here from this stage, from that same glucose molecule, you're going to get 36 ATP to make a grand total of 38 that's produced. Anabolism is where you actually sit there and build things up. It's a biosynthetic process. So the cells are actually going to build something. So the cells are going to sit there and actually create something from smaller molecules and manufacture. And a lot of times they use ATP to actually make this function of anabolism happen. One thing that also happens with anabolism is when we look at carbohydrates, we get something called dehydration synthesis. So this is where we can actually put multiple carbohydrates together, and actually what happens is they lose oxygen, they dehydrate, and they lose hydrogen too. So they pretty much lose water, comes out of it. And so you can make these polysaccharides just by removing a water molecule. And then, of course, to actually make them back into a monosaccharide, all you have to do is add water. So this is kind of a long explanation of catabolism and anabolism. And you can kind of see you have proteins here, carbohydrates that come in, they go into the glucose pathway, they become pyruvic acid. Acetyl-CoA ends up going through the Krebs cycle. You have some proteins that go this way, but a lot of your amino acids and proteins actually go here, where they're going to actually build other proteins, and then a waste product that's removed is going to be urea. Lipids are actually going to be put and stored back as fats, but they can also enter the pathway right here and turn into sugar, and also fire off the Krebs cycle. In the Krebs cycle, you're going to get carbon dioxide out, which we know, we talked about that mixes with water and can become bicarbonate as a buffer. Down here, you're actually going to get water produced through the Krebs cycle, is going to be excreted. You're also going to get things like lactic acid and produced right here. And the thing that happens is this is part of what we call the electron transport chain. One of the things that happens is this electron transport chain actually turns ADP, which is adenosine diphosphate, into adenosine triphosphate, which is the energy source of the whole body. Control of metabolic reactions. The main thing that ends up controlling them is enzymes, and so enzymes are products that actually help chemical reactions to occur, and so they work as a catalyst. So one of the things that ends up happening with multi-enzyme sequences of events of metabolic reactions is that one enzyme produces one chemical in a step of a process, and usually there's a multi-step process to get to the substrate, to the end substance that's actually being made. The activity depends on the molecular shape of the enzyme, and there's what they call an active site. So this is the region of the enzyme that actually binds to the substrate. Catalysts actually speed up the energy, and they do this by actually reducing the activation energy. So if something takes, say, a temperature of 100 degrees Celsius to occur, it can occur at 37 degrees Celsius, which is near body temperature, through a catalyst. The other thing they can do is catalysts can actually just speed up reactions. So if a reaction is able to happen only one time a second, a catalyst inside of an enzyme can actually speed that reaction up to where maybe it can now happen at 100 times per second. And so a lot of times catalysts and enzymes work hand in hand, and a lot of enzymes end up being catalysts. Some different cofactors that end up coming along that control metabolic reactions are things like iron, zinc, and copper. And a good example of this, of a cofactor of a non-protein substance that helps out, is oxygen binding to iron and hemoglobin. This reaction happens very fast. There's actually the hemoglobin actually ends up acting as a catalyst to allow this to actually happen, and this reaction happens very fast. So this is kind of an example right here of what a coenzyme does. And you have a non-organic protein substance, or non-protein organic substance, sorry, and it's going to, this coenzyme is going to functionally change the shape of an enzyme. So you can see right here, here's the cofactor, here's the enzyme, and here's the substrate that it's trying to put together, the chemicals. And if you don't actually put this cofactor in right here, things like vitamins, or in this case a mineral, kind of what we're looking at is, it would go right here. Let's say like in the case of hemoglobin, if you put iron right here, you got oxygen molecules right here. By actually changing the shape of the heme and the globin, you actually make it to where there's one of two things. In the case of hemoglobin, the iron would actually bind to the oxygen molecules and actually hold them in place. Or in the case of something else, you could actually change the functional shape of the protein and make it to where it would lock and then actually bind multiple molecules together and make a new substance out of it, or be able to transport the substance in the case of oxygen and hemoglobin. One thing when we look at energy for these metabolic reactions, ATP is the main thing that is used to actually make these occur. There's some other factors that we can use too for metabolic reactions. There's something called NADH and FADH2, and a lot of times these are actually converted over into ATP. And so the main thing when we look at these molecular bonds and energy right here, adenosine triphosphate it's actually adenine right here, which you can remember is a function of nucleic acid. And you have the ribose sugar right here, so you've actually got to have DNA to make this, because with the ribose sugar, this is something like a strand of RNA. So you actually have to have DNA to actually make this adenine right here. And then what you end up doing is you end up putting on 3 of these phosphorus tails to make ATP. And when this third one breaks off, tremendous amount of energy is actually released, and it's enough to actually keep the body going. Of course, this energy has to be replenished, and how that happens is through fueling the Krebs cycle. So when we look at this, we've kind of talked about this with anabolism, but catabolism is going to occur in the cytoplasm, and originally it's going to be anaerobic. So out in the cytoplasm it's anaerobic, and glucose is broken down from a 6-carbon ring into 2 3-carbon chains of something called pyruvate or pyruvic acid. And this pyruvic acid is then transported to the mitochondria. When it enters the mitochondria, it's going to go through the Krebs cycle. And so from the Krebs cycle, it's going to produce additional ATP. So as you can see right here, glucose is going to come in, and it goes through this little reaction right here, and on its way to making 2 ATP, is going to end up making the pyruvic acid right here. And then it's going to go through the Krebs cycle. And through the Krebs cycle, it's going to come back, and in turn, in the something we call the electron transport chain, it's going to produce an extra 36 ATP. So when we're looking at this in cell metabolism, the most important thing to understand out of all this is, and this right here is a little bit confusing because it has the number of ATP wrong out here in the cytoplasm with the pyruvate. So that's something to understand, is when you're looking right here, some of this actually doesn't quite make sense. If you actually look on your book on page 305, that is the more correct way that everything goes. They have too many ATP up here on this little chart, so that's why I'm skipping by it. But what's important to know is that you get 2 ATP that come off of glycolysis, and each molecule of pyruvate that ends up running through here produces an additional 18 ATP. But there's 2 molecules of pyruvate made. So that's why we end up saying 36. So an additional 36 are made. And this is both through the Krebs cycle and something called the electron transport chain. So those things that we talked about earlier of NADH and FADH2, those actually go through the electron transport chain. And so these electrons actually hit ADP and a phosphate molecule, and they actually make them bind together into ATP. And so you can look through, it's a very confusing thing when we look here at the Krebs cycle, understandably so. You can actually take a 5-hour course just on this that's all semester long. So I'm trying to make it as simple as possible. Trust me, I had the 5-hour course all semester long. So there's a lot of steps that go along the way. And once again, it's good to understand what the Krebs cycle does. I don't expect you to know that it becomes citric acid to isocitric acid to ketoglutaric acid, and that NADH is given off here, and NADH here, and FADH2 and all this. What you need to know is that glucose is broken down into pyruvic acid from glycolysis out in the cytoplasm, which is anaerobic. This is how all anaerobic bacteria live. And the huge step in evolution for us is aerobic metabolism in the mitochondria, where all at once these byproducts of breaking down glucose are run through, and instead of just getting 2 ATP, an extra 36 are fired all off, and it all comes from oxygen. Some of the byproducts off is going to be lactic acid. Other byproducts that are going to come off of this are actually going to be carbon dioxide. What happens with the electron transport chain is you've actually got these different hydrogen molecules out here with a positive charge. There's a negative charge on the inner core. And so what actually happens is the negative charges are actually forced across with these hydrogen ions, and they actually lose an electron. And when they lose an electron, energy is actually transferred across this membrane, and that electron is shot back. And it actually comes back in here and keeps the negative charge on the inside of the mitochondria, to where there's always extra electrons in here, and they're always trying to make it across to this positive side, but they get trapped. And what happens is they get shot back, and ADP is then turned into ATP. And this is all fired once again from that NADH and that FADH2. So this is what the net result ends up being, is that out of this, glycolysis only gives 2 ATP, which is not bad energy off of glucose. And this is what anaerobic bacteria is able to do by itself. But this is the beauty of aerobic metabolism. As you count it up, there's an additional 36 ATP, meaning that totally out of a molecule of glucose, we go from anaerobic, which you only get 2 ATP, to with aerobic metabolism, now we have 38. So we keep the original 2 and we add 36 to it from that same molecule of glucose. Lipid metabolism. In lipid metabolism, we have something called hydrolyzation. So triglycerides are hydrolyzed into glycerol and 3-chain fatty acids. So this actually means that when something's hydrolyzed, that hydrogen ions are lost. And so we can actually take those fatty acids that are long-chain carbons. Well, pretty much when they go through the liver, they're changed from being plant sources from unsaturated to saturated sources. And those glycerol molecules and the fatty acids themselves are actually broken down and can be broken down into basically glucose and run back through as the acetyl-CoA enzyme gets them as pyruvate and runs them back through the Krebs cycle. And they can produce ATP. One thing off of this energy of the Krebs cycle too also that's produced is whenever you have something like this and energy is being changed, you always do have some heat production. But fatty acids, the glycerols easily are, if you remember, a sugar chain backbone, they're easily converted over into glucose. Fatty acids themselves can be, and fatty acids can also make ketone bodies, and these ketone bodies can then actually be converted over into glucose also. One of the things that happens sometimes is like in diabetics, the body actually goes through this and starts breaking down fat trying to get to the sugar, and the glucose is already high, but the cells aren't getting any, and the cells say, hey, we need it for the Krebs cycle. And so you'll notice with ketoacidosis, what happens is the fats in the body start to break down, and they overproduce ketone bodies, and so that's where we get ketoacidosis from. And to some extent, ketone bodies can actually make it into the cell and can help to drive this Krebs cycle. But the rest of the glucose and the rest of the glycerol and the rest of the fatty acids are actually kept outside the cells. In protein, we have amino acid catabolism, and this happens at the mucosal level. The kidney can do it, the brain, the liver, the skeletal muscle. Amino acids actually go through what they call deamination, or transamination. With deamination, what actually happens is that the nitrogen base is actually broken down in it, and you actually get ammonia produced. And this ammonia is going to be then changed by the liver over into urea and excreted from the body. In transamination, you're actually just moving your nitrogen base from one amino acid to the other. So here's kind of one of these things of transaminase right here, to where you have right here a nitrogen base, and it's what they call glutamic acid, and this is a keto acid right here. And then you can actually take this glutamic acid right here, and you can actually move the nitrogen over to this keto acid. And then right here you get another keto acid that was glutamic acid, and this that was just a keto acid now becomes tyrosine. And so the body does this a lot of times to make its own amino acids, and this is how it actually does it. It's just by moving that nitrogenous base from one acid to the other to actually make a new amino acid. The other thing it can do is it can actually take and remove the nitrogen base. And so you just get a keto acid, which the body can then turn into glucose, and then you get ammonia that's given off. And this ammonia is very toxic to the body, so what the liver does is it actually takes, when it's functioning normally, it takes all this nitrogen, binds it together, and actually makes urea that's going to be excreted from the body. So that's it for Chapter 12 on nutrients and metabolism.